When I think of a protein, at least the ones that I study, I think of a little machine, like literally a 2, 3, 4 nanometer size machine that's evolved to do these really complicated things. We're looking at the spatial organization of dopamine receptors. And the open question, the thing that people don't know, is whether these proteins actually touch. Do they actually bind each other and interact? If they interact directly, the idea is that we could make a drug that actually breaks up the interaction. Then you can treat new disorders. And then it'll also open up new roads for new treatments.
My role is performing the experiments, collecting the data, and working to interpret the results.
So this technique relies on fluorescence. So when we're actually doing the measurements, the room lights have to be off, the curtains have to be closed, because otherwise the detectors will get flooded with room light. The reason we need this equipment is because we first have to zoom in and isolate one single cell. And then we have to use pulsed lasers to look at a tiny region of that cell.
So the microscope itself, that's commercially available, but then like all the things that are involved in the whole setup that we use to collect the data, a lot of that's custom built.
So the microscope itself is to look at the cell, the lasers, and all the electronics behind me, those are to look at the proteins within the cell.
It goes to these detectors, which gets read here, and then is transferred to the computer where we can visualize the data that we're getting. If you were to take a human hair, that's 80,000 nanometers, and then this protein that we're looking at is on the order of like 2 nanometers.
They're tiny. They're molecules.
There's a lot of experience gained from just understanding and learning how to work with these types of tools.
The thing I like best about the research is that I not only get to use cool toys, but I get to try to solve important problems.